Our group is mainly interested in understanding how the immune system functions. And we are particularly interested in studying a particular type of cells of the immune system called lymphocytes. And lymphocytes are essential for our body because they produce factors that are called antibodies. And antibodies are actually binding to viruses and bacteria as soon as we get in contact with them. And in the moment they interact with antibodies, these bacteria are neutralized by our immune system, and therefore we're able to defend ourselves from the external world. So during an immune response, B lymphocytes recognize viruses and bacteria. In doing so, they actually proliferate very intensively. And during this proliferation time, or during this proliferation stage, lymphocytes do introduce mutations in their genome. So this is a very unique feature of lymphocytes. These mutations are thought to increase and improve the affinity of antibodies against viruses. However, sometimes the machinery does mistakes and introduces unwanted mutations in other regions of the genome. Now, if you're hitting a gene that is involved and is regulating cell division or prevents cell death, you get a cancer called lymphoma. Now, lymphomas are the most common type of cancers in the Western world among at least the blood cancers. And these tumors are, to a large extent, fairly well treated at the time, but few of them are still remain deadly. So we need still to identify better therapies that would actually allow these forms of deadly tumors to be still and efficiently treated. Now, the mission of my lab is, on one hand, to understand which genes are mutated during an immune response that lead to the development of cancer, and on the other hand, to identify drugs and potential ways to interfere with the function of these genes, thereby finally trying to find new cures for the treatment of some of these deadly diseases that cannot to date still be treated. In the course of identifying genes that are relevant for cancer, we got very interested in a group of proteins called polycomb group proteins that regulate over 6,000 genes in our genome. Now, among those genes, several are actually important in the regulation of cell division, and we have found that actually they play an important role during a normal immune response in B lymphocytes. Moreover, in the process of understanding how polycomb proteins regulate cell differentiation and survival, we actually discovered that these polycomb proteins are able to play an important role in reprogramming somatic cells, rendering similar to stem cells. Now, this is a process that is very similar to what happens in cancer when cells lose their identity and become stem cell-like. So we think that through the identification of these polycomb target genes that we have discovered through this study, we can now try also to understand how polycomb contributes to lymphoma progression and rendering these cells immortal. Luckily, there have been in the last year a number of papers showing the identification of small molecule inhibitors that actually interfere with polycomb proteins. So we are very eager to try this in preclinical models and to test whether they are indeed efficient to treat these deadly lymphomas that actually deregulate the activity of these polycomb proteins in a very frequent manner. We have in the past years identified a number of genes that are mutated very commonly in lymphomas. However, we don't really know how these genes work. We know that they are mutated, but we don't know how they impact on the development of the disease, on metastasis of the disease. So we are now interested in entering in a new stage where once having identified these genes, we have to start to study their function and how they participate at specific stages of tumorigenesis. At the same time, we have identified genes that are mutated in cancer, in lymphomas in particular, and we are now using this information in collaboration with oncologists in the clinics to see whether some of these genes can be used as diagnostic markers or markers that can allow us to predict in a better way the response of patients to new treatment.